Hello everyone. Today we will discuss the second genus of gymnosperm, that is Pinus, and I will cover it in two parts. Today we will discuss about its external morphology, some internal structures, and the species that we will focus on is Pinus roxburghi, commonly known as chair pine. Our today's learning objectives are: we will discuss about its occurrence, ये कहाँ पे पाया जाता है, and its external morphology, internal structures, that is internal structure of root, stem, and leaves, and especially we'll discuss about the internal structure of Pinus needle. So today we will discuss these topics in Pinus. Before discussing this, we will start with the systematic position of Pinus roxburghi. So it belongs to division Gymnospermae, and its class is Coniphyropsida because its external appearance is conical, so that's why it is in class Coniphyropsida, and it belongs to order Coniferales. Family is Pinaceae, and genus is Pinus, and species is roxburghi, commonly known as chair pine. Occurrence: Pinus roxburghi is commonly known as common pine or chair pine. It is mainly distributed in the northern hemisphere to the subalpine region and is represented by about 114 species throughout the world. In India, there are 6 species of Pinus which have been reported, out of which 2 are very common. One is Pinus longifolia, also known as Pinus roxburghi, that is the common pine, and Pinus excelsa, or Pinus, its synonym is also Pinus vallicana, commonly known as blue pine. So this is Pinus roxburghi, and this is Pinus excelsa or Pinus vallicana, commonly known as blue pine because it shows slight bluish tinge. Pinus roxburghi. It is commonly found in the hills of Kashmir, Himachal Pradesh, Punjab, and UP at an altitude between 420 to 2250 meters from sea level. So this is Pinus roxburghi, and it is commonly known as chair pine, and it attains a height of about 30 to 50 meters. So ये इसका height ये attain करता है, and its foliage leaves are long, needle shaped, trifoliar, that we'll discuss in upcoming slides, and triangular in cross section. So this is the unique feature of this Pinus roxburghi. Next, these are species which are found in India. Next species is Pinus gerardiana, commonly known as चिलगोजा pine. This is the female cone, और इस female cone के अंदर होते हैं seeds, and these seeds are dry. These seeds are edible, which are commonly known as chilgoza. So this is commonly known as chilgoza pine, and it is in Himachal Pradesh. It is mostly found in Kinnaur region. It is found in altitude ranging from 2200 to 3300 meters from sea level, and reach up to the height of 10 to 20 meters. And foliage leaves are long, trifoliar or triangular, similar to that of Pinus roxburghi, and seeds are 2 to 2.5 centimeters in length and are used as delicious dry fruits. So these are the seeds of chilgoza, pine nuts, chilgoza, and this is the packing of these fruits. They are rich in nutrients. So from these cones, these seeds are collected and are dried and are rich in various nutrients and minerals. Next is Pinus excelsa, commonly known as Pinus vallicana. It is also known as blue pine. So, or in Hindi, it is also known as cal. So it is commonly known as silver fir, blue pine, or cal. It grows in the hilly regions at an altitude ranging from 1200 to 3500 meters from sea level, and foliage leaves are bluish, so that's why this is known as blue pine, and are pentafoliar. It means it contains 5 needles. And female strobili are cylindrical. So these are the photographs of these female cones of Pinus vallicana. Pinus insularis, commonly known as Khasi pine because it is found in the Khasi hills of Assam. This is the photograph of Pinus insularis. It is similar to that of Pinus roxburghi. And these are the female cones of Pinus insularis. So it is commonly known as Khasi pine and distributed in the Khasi Naga hills of Assam and Manipur, and it occurs in the altitude range of 700 to 1850 meters from sea level and may reach up to the height of 30 meters. Its foliage leaves are needle-like and are trifoliate, similar to that of Pinus roxburghi and Pinus gerardiana, and this is the good source of timber and resin. Pinus mercussi, commonly known as Marcus pine, and this is the photograph of this Marcus pine. It is commonly found in West Bengal and is found at a low altitude of 150 to 600 meters from sea level, and it may reach up to the height of 30 meters. And its foliage leaves are trifoliar. It means it bears 3 needle-like structures, and it is the most common conifer of Siam. And this is the female cone of this Marcus pine, and it is trifoliar in nature. It bears 3 needle-like structures. Next is Pinus armandi. It is commonly known as almond pine, and this is the photograph of this almond pine. It is commonly known as almond pine, and it occurs in the Northeast Frontier area and is also abbreviated as Northeast Frontier Agency, which include the area of Arunachal Pradesh and some part of Assam. And it is found at an altitude of 1500 meters from sea level. And this is the female cone of this almond pine, and it attains a height of about 15 to 18 meters, and its foliage leaves are pentafoliate, which is similar to Pinus vallicana. Next is external morphology of Pinus roxburghi, and as we can see in this photograph, it is long, erect, perennial evergreen tree in which the branches are arranged in spiral fashion. So this is tall, erect, perennial evergreen tree with a spirally round branches, and it appears pyramidal or conical when it is young and rounded or flat topped with age. And the plant body is sporophytic in nature because it means it is diploid. Next comes root system. In case of Pinus, it has well developed taproot system which is formed by the radicle of embryo. And this photograph we can see here, this is the extensive root system which is formed by this Pinus. From this main taproot there develops number of lateral roots. So it shows taproot system, and this taproot system gives rise to extensive lateral roots which fix the plant body firmly or strongly to the soil surface. And in these roots, root hairs are scanty or very less, and this function of root hair is taken up by fungal mycelia which forms the ectotrophic mycorrhiza. Ectotrophic means that it shows this symbiosis or association with the surface of roots only, and this ectotrophic mycorrhiza helps in absorption of minerals and nutrients. It also helps in the absorption of water. So in this photograph we can see here, this is the extensive root system which is formed by the Pinus, and it shows association with the fungal mycelium to form ectotrophic mycorrhiza. And this helps in absorption of nutrients and also protect it from pathogens. And mostly members of Basidiomycetes like Amanita, Boletus and Rhizopogons are found in association with the roots of higher plants, that is roots of Pinus, to form mycorrhiza. And the normal roots are long. In case of Pinus, the normal roots are long, thin and has root hairs but lack root caps and fungal hyphae. So जो normal roots है उसमें fungal association show नहीं करते हैं, and they lack root caps, otherwise they have root hairs. Next comes stem. So in this photograph you can see here it is erect, woody. So it is erect, tall, cylindrical, woody and branched. And branching जो होती है इसमें, that is monopodial, and lower branches are longer and horizontal, give the conical shape to the plant. जो lower branches होती हैं वो larger size की होती हैं, लंबी होती हैं, and they give conical appearance to the plant. And branches are dimorphic, means having two types of branching. One are long branches with unlimited growth because they have apical bud in these branches, and second one are dwarf shoots, which are commonly known as spur or brachioblast, with the limited growth, means they are the branches of limited growth. So in this photograph we can see here, so this is the long shoot, this main shoot is the long shoot or branch of unlimited growth, and this one is the dwarf shoot, which is the branch of limited growth. The next is branches of unlimited growth or long shoot. So this branch is the branch of unlimited growth because it bears the apical bud in its tip, or apical meristem in its tip. So these possesses apical bud and grow for unlimited period and are called long shoots, and also known as branches of unlimited growth. And branches are spirally arranged on the main trunk that we have already discussed, and these also bear scale leaves and dwarf shoots on the axil of the scale leaves. So these are the dwarf shoots. These smaller are the dwarf shoots which are borne on the axil of scale leaves, and older portion bears the scars of the dwarf shoots. जो older portions होते हैं उसमें scars हो जाते हैं dwarf shoots के. Next comes branches of limited growth or dwarf shoots. ये जो branches होती हैं इसमें apical bud present नहीं होता है. So these are the branches, these smaller ones are the branches of limited growth, and they are borne on the axil of scaly leaves. So these lack apical bud and grow for the shorter period, and these are also known as spur shoots or brachioblast. इनको spur shoot और brachioblast भी कहा जाता है, and they arise in the axils of scale leaves on the long shoots. These consist of 8 to 10 spirally arranged scale leaves which are known as cataphyls. So these scale leaves are also known as cataphyls. So इसमें बाहर जो scaly leaves हैं इसको बाहर से cover करती हैं. They are 8 to 10 in number और spirally coiled होती हैं. And these scale leaves are also known as cataphyls and terminates into 1 to 5 needle-like foliage leaves, और बाद में ये turn हो जाती है into needle-like foliage leaves at its apex, और इसके apex से ये needle-like structures निकल. Next comes leaves. So leaves भी dimorphic होते हैं, that we have already discussed. One are scale leaves and other are foliage leaves. First is scale leaves. So these are thin, brown, flattened and minute in structure. So ये जो structures हैं, these are the scale leaves and are borne on the long shoots as well as on the dwarf shoots. ये दोनों में present होते हैं, long shoot में present है और dwarf shoot में present है, and have distinct midrib और इसमें distinct midrib present होती है. और in these scale leaves are also known as cataphyls. इनको cataphyl भी कहा जाता है. And इनका जो function है, it protects birds and conserve water around the branches. So they helps in conservation of water. Next is foliage leaves. So these are green in color, which are acicular or needle-like in its structure. So these are borne on dwarf shoots, and these are long, acicular, needle-like and green in color. They have lifespan of 3 to 10 years, and they are responsible for the evergreen nature of the pinus tree. So they remain green for a number of years, that is 3 to 10 years, and hence the plants are evergreen. And the number of needles per spur varies from 1 to 5, and this number of needle character helps in differentiation of various species of pinus. So we have monofoliar leaves, it means having single needle-like structure, which is found in case of Pinus monophylla. Then we have bifoliar leaves having two needle-like structures, that is found in case of Pinus mercosi and Pinus sylvestris. Next is trifoliar, having three needle-like structures in case of Pinus roxburghi, Pinus gerardiana and Pinus insularis. Then we have quadrifoliar leaves having four needle-like structure, it is found in case of Pinus cordifolia. Then we have pentafoliar leaves having five needle-like structure, it is found in case of Pinus excelsa or Pinus vallicana and Pinus armandi. And this slide shows the various diagrams of various types of needles or leaves, foliage leaves, which are found in various species of Pinus. So this first one is of Pinus monophylla, it means having single needle-like structure. Then second one is of Pinus sylvestris, having two needle-like structures. Then third one is of Pinus roxburghi, having three needle-like structures. Fourth one is of Pinus cordifolia, having four needle-like structures. And last one is of Pinus valiciana or Pinus excelsa, also known as blue pine or cal, having. Next, we will discuss about the internal structures in case of Pinus. Firstly, we'll discuss about the anatomy of root. So, anatomy of root is just identical to that of dicotyledonous root. It shows various parts. The outermost layer is epidermis or epiblema. It is made up of parenchymatous cells, which is a single layer thick outermost layer, and the sum of the parenchymatous cell give rise to root hairs. Then this epidermis is followed by a broad zone of cortex, which is made up of parenchymatous cells, and innermost layer of cortex forms anodermis, which contains prominent Casparian strips. Then this anodermis is followed by 6 to 7 layers of pericycle, which are again made up of thin-walled cells. Inside this pericycle, there is presence of vascular bundles, which consist of xylem and phloem. And as we know, in roots, the vascular bundles are radial. So in Pinus root, the vascular bundles are also radial. So outermost layer is epidermis. It is made up of single layer of parenchymatous cells with some unicellular hairs, which are present only in the young roots. Then this epidermis is followed by many layers of thin-walled parenchymatous cortex. This innermost layer of cortex is the endodermis, and it contains prominent Casparian strips that helps in formation of a watertight channel. Then this endodermis is followed by 6 to 7 layers of thin-walled pericycle, and it mainly contains tannins and starch. Then this pericycle is followed by vascular bundles, and vascular bundles in case of roots are radially arranged, and they are diarch, means having 2 arms, 2 rays, to hexarch, having 6 rays of protoxylem, with exarch protoxylem. Exarch means when the protoxylem faces towards pericycle. This protoxylem is bifurcated or Y-shaped, that we can see in this diagram, towards periphery, and in between these bifurcation there is presence of resin canal. So in इस bifurcation के बीच में resin canal present होती है, that we can see in this diagram also. And phloem is present alternate to protoxylem. Protoxylem के साथ ही phloem present होता है alternatively, and this pith in case of root is poorly developed or either absent. So this is the anatomy of young. Next is the secondary growth in case of root. So tiers of old root showing secondary growth. As we know that secondary growth is the growth in girth or growth in diameter. So this growth results in enhanced or increased diameter of the root. So this is the tiers of old root. This secondary growth starts with the conversion of some parenchymatous cells in between xylem and phloem, which becomes meristematic, and they form the cambium. This cambium results in formation of secondary xylem cells towards inner side and secondary phloem cell towards outer side. In addition to this, during secondary growth इसी टाइम पे एक और कैम्बियम की रिंग बनती है in the outer pericycle, which is the cork cambium, and this cork cambium cuts parenchymatous cells towards inner side, जिनको बोला जाता है जो बनाते हैं secondary cortex, which are parenchymatous cells, and towards outer side it will form the thick layer of dead cells, which is known as cork. So cork, also known as phallum, and cork cambium, also known as phallogen, and secondary cortex, which is known as phalloderm. These three combine to form one structure which is known as periderm. So these are some events which occur during secondary growth. In addition to this, जहाँ पे ये resin canals present होती है, वहाँ पे ये cambium cut करता है parenchymatous cells की rays which are known as medullary rays. So ये medullary rays भी present होती है in the region of secondary xylem. So many uniseriate means single cell thick medullary rays are present in the region of secondary xylem. And primary phloem, जो primary phloem है जैसे ही secondary phloem बनता है it gets crushed और ये crush हो जाता है and secondary phloem exists. Then in the outer region that I have already discussed, जो outer layer है पेरिसाइकिल की वहाँ पे कुछ एक cells मेरिस्टेमेटिक बन जाते हैं. They form the cork cambium and this cork cambium is also known as phallogen. It is formed in the outer region of this pericycle, and this cork cambium or phallogen cuts secondary cortex towards inner side and cork cells externally towards outer side, and these three combines to form periderm. This primary xylem is the same as in young roots. जैसे young root में primary xylem था वैसा ही इसमें present होता है. Each group is bifurcated Y-shaped structure और इसके बीच में present होता है resin canal and between the bifurcation. So this is all about the secondary growth in case of pinus root. Next comes anatomy of stem. So anatomy of stem is again identical to that of dicotyledonous stem. इसमें सबसे बाहर वाली लेयर होती है epidermis. Then this epidermis is heavily cuticularized. It is followed by a multi-layer cortex. और जो outer layer है cortex की that is hypodermis, it is sclerenchymatous. Then inner cells are parenchymatous. और ये जो inner cells हैं parenchymatous they contain multiple resin canals. Then this innermost layer of cortex is endodermis. It is single layered, which is followed by multi-layer pericycle. Then inside the pericycle there are multiple vascular bundles which are conjoint, collateral, open and endoc. So it is similar to that of dicotyledonous stem, appears wavy in outline and is differentiated into epidermis, which is the outermost layer, which is single layered and thick walled cell present होते हैं इसमें. Epidermis is heavily cuticularized, बाहर से cuticle की heavy covering होती है. The next is cortex. Epidermis को follow क्या करता है multi-layered cortex. इसमें जो outer layer है cortex की they are sclerenchymatous, there we can see in this diagram, and some inner layers are parenchymatous. और जो inner layer है parenchymatous cortex की इसमें बहुत सारे resin canals present होती हैं. So in the inner layers of cortex are present many resin canals. Then cortex is followed by a single layer endodermis and multi-layered pericycle. Next comes vascular bundles. जो vascular bundles हैं they are conjoint, collateral, open and endoc. There are approximately 5 to 10 vascular bundles which are arranged in the form of ring. So इसमें हम देख सकते हैं this is single vascular bundle. So इसकी तरह 5 से 10 vascular bundle present होते हैं और ये arrange होते हैं ring की form में. And cambium ring is present that we can see. So that's why these vascular bundles are open in between xylem and phloem. And phloem consists of sieve cells. इसमें sieve cells present होते हैं, sieve tubes present नहीं होती हैं. And sieve plates are there. And phloem parenchyma is also there, but the companion cells are lacking. Companion cells इसमें absent होते हैं and some albuminous cells are present. Next companion cells इसमें absent होते हैं. Then pith is small parenchymatous and is present in the center of stem. So this is all about the anatomy of young stem of pinus. Next is secondary growth in case of stem, pinus stem. So it begins with the formation of two lateral meristems, which is just similar to that of the dicotyledonous stem. So one is the intrafascicular cambium, which is formed between the vascular bundles that will form a complete ring, और वो जो complete ring है cambium की that will cut towards inner side the secondary xylem and towards outer side the secondary phloem. While in the outer region of cortex there is formation of another cambium which is known as the cork cambium or phallogen. It will produces cork cell towards outer side and secondary cortex towards the inner side. And these three things, that is cork, also known as phallum, phallogen, that is cork cambium, and the phalloderm, which is the secondary cortex, these three things combine to form the periderm. So what is secondary growth? It is the growth in girth or increase in diameter due to the activity of lateral meristem. So two lateral meristems in conifers are one is vascular cambium जो बनाता है secondary xylem and secondary phloem, while the second is the cork cambium. और ये जो second cambium है cork cambium, it produces the cork cell towards outer side and secondary cortex towards the inner side. This interfascicular cambium develops from the parenchyma cells of medullary rays adjacent to the intrafascicular cambium and combine to form a complete ring of vascular cambium. So जो दो vascular bundles हैं उनके बीच में जो parenchyma cells हैं medullary rays के, they becomes meristematic and they will form a complete ring of cambium. And this ring is vascular cambium, which is formed by the combination of intrafascicular cambium, that is within the vascular bundle, and between the vascular bundle, that is interfascicular cambium. So these both cambiums combine to form vascular cambium that cuts secondary xylem towards inner side and secondary phloem towards outer side. So it cuts secondary xylem towards toward inner side and secondary phloem towards the outer side. Next, in secondary xylem, annual rings of thin-walled spring wood is formed. जो secondary xylem है, in spring season there is abundance of food material. तो जो xylem के cells हैं वो large size के होते हैं and they are lighter in color, and that band of secondary xylem forms the spring wood because it is found in the spring season when there is abundance of food material and water in the soil. And then in winters or autumn, thick-walled wood is formed because there is scarcity of food material and water. So that is formed in autumn season, so that is the autumn wood. So these both spring wood and autumn wood, these combine to form one annual ring. And this demarcation of these rings is very clear in case of temperate trees. So such compact wood is known as pycnoxylic wood. So this is the speciality of the spinous wood, while in case of cycas we have discussed that wood is menoxylic wood, which is the spongy wood. So we can calculate the age of tree by calculating the number of annual rings, and this technique of calculating the age of tree by counting annual ring is known as dendrochronology. So we can calculate the age of tree by counting these annual rings. Below the secondary xylem, there are present a few groups of endark primary xylem. जो secondary xylem है उसके नीचे present होता है endark primary xylem, which contains the resin canals. Then the central part of stem is filled with the parenchymatous pith. The central part is occupied by parenchymatous pith. Then in addition to this, resin canals are also present, which are distributed in secondary cortex. Cortex में भी present होती है resin canal. They are also present in the secondary xylem and secondary phloem, and rarely in case of pith. So these resin canals are distributed throughout the tissue. While there are some medullary rays, जो medullary rays हैं जो connect करती है pith को cortex के साथ. So these medullary rays are known as primary medullary rays. While जो medullary rays बनती हैं secondary xylem और secondary phloem के बीच में, these are known as secondary medullary rays. So primary medullary rays are the rays which connect this pith with the cortex, while secondary medullary rays are present in the secondary xylem and secondary phloem. So resin canals are present in cortex, secondary xylem, primary xylem, and rarely in pith. So this is all about the secondary growth in case of pinus stem. Next comes one important concept, that is sectioning of cylindrical organs in plant. Cylindrical organs are like stem or roots. So we can cut various sections through various planes, so that we'll understand from this slide. So this is the image which shows various sectioning through various planes. So first we have transverse section. So transverse section is when the organ, that is stem or root, is cut right angle to the longitudinal axis. So this is the longitudinal axis, this one. If we cut the section right angles to this longitudinal section, so that section is known as transverse section or TS. If we cut the section right angles to the transverse axis, this is the transverse axis, and we cut the section right angle to this transverse axis, then the section is known as LS. that is longitudinal section. This longitudinal section, there are two sections which are possible in this plan, that is longitudinal section. One is this transverse longitudinal section. It is the section in which the section is cut along one of its tangent. जब हम किसी एक tangent से longitudinal की तरफ इसका section काटते हैं, so this image shows if we cut this section through this plane, that is one of its tangent with the help of razor, so that section is known as transverse longitudinal section. Then next comes a radial longitudinal section. If we cut the section through one of its radii, तो किसी एक radii से जब हम इसका section काटते हैं, then that section is known as radial longitudinal section. We have some dorsiventral organs, like we have leaf thallus of liverwort, that is Mertensia or Rixia. Then we so in this case we can cut the section through this plane, and this plane is known as vertical transverse section. So these are various planes of sectioning in case of plant organs. So in Pinus we will also discuss about this TLS and radial longitudinal section in upcoming slide. Next is radial longitudinal section of Pinus wood, also abbreviated as RLS, when the section is cut along the radius, and in this section pith is also visible. So when the section is cut through this region that is long, which is through one radius, so this is called a radial longitudinal section, and it shows that there are the parenchyma cells which are composed of two types of cells. One is the central living cells called ray parenchyma, and peripheral dead cells जिनको कहा जाता है marginal tracheids, that we can see in this diagram. So these marginal cells are the tracheid cells centrally. These are the ray parenchymatous cells which store starch, and these tracheidal cells contain border pits which helps in exchange of material. So peripheral tracheids have border pits, and these border pits have special thickenings which are known as bars of sinew. So border pits are surrounded by bars of sinew which are made up of cellulosic and pectin substances. जो ये thickenings हैं, these thickenings are known as bars of sinew, and they are made up of cellulose and pectin in tracheids, which are seen on the surface view. So this is the photograph which shows these bars of sinew which surround the tracheids, and these are the thickenings which are made up of cellulose and pectin. An area of contact between ray cells and tracheids are called cross-field. जो area है ray parenchyma के बीच में और tracheidal cells के बीच में, उसको कहा जाता है cross-field, and is marked by simple pits. इसमें present होते हैं simple pits. In the xylem region, thick medullary ray cells are surrounded by tracheids. जो xylem region है वहाँ पे जो medullary cells हैं, they are surrounded by the ray tracheids. Then the thin-walled ray parenchyma is also present. इसमें ray parenchyma भी present होता है. So this is the medullary ray, and these are the albuminous cells. These medullary ray cells also store starch. Then the xylem is separated from phloem with the help of cambium. जो xylem और phloem है ये एक दूसरे से separate होते हैं with the help of this cambium, which is the meristematic tissue. And albuminous cells are present in the medullary ray region that we can see, and also in the phloem region. So albuminous cells present होते हैं medullary ray में, in the phloem region. जो phloem region है वहाँ present होते हैं, and phloem consists of sieve tubes, sieve plates and phloem parenchyma. इसमें जो companion cells हैं absent होते हैं, so it consists of the sieve cells, we can say, and sieve plates and phloem parenchyma. And in this case pith is absent, pith is present. Sorry. जब हम RLS स्टडी करते हैं, so pith is visible. In case of tangential longitudinal section, pith is not visible. So this is all about the RLS of Pinus wood. Next comes tangential longitudinal section, abbreviated as TLS of wood. When we cut the section through one of the tangent of wood log, so that section is known as tangential section. So this image shows the TLS of this wood log, when we cut this section through this area, this is known as the tangential surface, and when we cut the section through this, and this is known as TLS. And it shows the height and width of ray. जो ray parenchyma है उसकी ये height और width बताता है. And following structures are visible, which shows the border pits and medullary rays. So these two structures that are border pits और medullary rays visible होते हैं इस section में. And each border pit, जो border pit है, that is enclosed by pit chamber. Pit chamber होता है bounded by pit membrane and contains centrally located swollen structure जिसको कहा जाता है torus. So इसमें एक swollen structure present होता है center में जिसको कहा जाता है torus. So this is the border pit and this centrally located structure is known as torus. Tracheids are composed of rectangular cells. Tracheids बने होते हैं rectangular cells से. So this is again the photograph which shows this radial longitudinal section. Middle lamella इसमें बहुत clear होता है. And there are many uniseriate medullary rays, means single cell thick medullary rays present होती हैं इसमें. In the xylem region, medullary rays contain central located starch cell. जो xylem region है वहाँ पे medullary rays store करती हैं starch and are surrounded by tracheidal cells, और उनको surround करते हैं tracheidal cells. And albuminous cells भी present होते हैं इसमें in the medullary ray in the phloem region as similar to that of the radial longitudinal section. और इस केस में जो pith होती है that is absent. Pith इसमें visible नहीं होती है. So this is the difference. In case of RLS, pith was visible. In this case, that is TLS, pith is not present. Next comes the internal structure of dwarf shoot. It is just similar to that of the long shoot. It shows various structures. It consists of outermost epidermis, which is made up of thick walled cells and is covered by cuticle. Then this epidermis is followed by cortical cells, cortex cells. इसमें भी जो outer layer है cortex की, that is made up of thick walled cells and contains various numerous resin canals which are distributed in the cortical region. इसमें जो endodermis और pericycle है, they are not distinct, clearly demarcate नहीं कर सकते हैं उनको. Then this also contain vascular bundles, और जो vascular bundles हैं, they are just similar to that of the vascular bundles of long shoot. So it consists of outermost layer which is epidermis composed of thick walled cells which are cuticularized. Then it is followed by the cortex, और इसकी जो outer layer है that is lignified or made up of sclerenchyma, that are dead cells. Then it is followed by thin walled parenchymatous cells. Resin canals scattered होती हैं cortex में and endodermis और pericycle इसमें distinct नहीं होती. So these two layers are not distinct. Then vascular tissue की बात करते हैं. So it is just similar to that of the long shoot. They are arranged in rings. और इसमें जो number है vascular bundles का, that is generally 6. So we can see here these are 6 vascular bundles. However, ये जो number है, it can vary from species to species in case of Pinus dwarf shoot. Then the structure of vascular bundles is similar to that of the long shoot. That is, it contains conjoint, collateral, open and endark vascular bundles. And the secondary growth is limited in case of dwarf shoot. Pith of dwarf shoot is comparatively smaller. Pith इसमें small होता है size में as compared to the long shoot. And dwarf shoot ends into the variable number of foliage leaf that we have already discussed. That number may be 1, 2, 3, 4 or 5. So this is all about the internal structure of dwarf shoot. Next comes the TS of needle, or we can say foliage leaf. So this is the section of Pinus roxburghi under microscope. Its shape also varies from species to species. So it is circular in outline in case of Pinus monophylla, having single foliage leaf. Then it may be semicircular in case of Pinus sylvestris, and it may be triangular in case of Pinus longifolia and Pinus roxburghi. So we will study only the TS needle of Pinus roxburghi. So it shows outermost layer which is epidermis. It is the outermost layer which is made up of thick walled cells. It is covered by the cuticle, heavy cuticle, and there are many sunken stomatas which interrupt this epidermis, and these stomatas are of haplochelic type. Haplochelic type stomata means जब subsidiary cells और जो guard cells हैं they are not derived from the same cells. Same mother cells से नहीं बनते हैं subsidiary cells or guard cells. So that type of stomata is known as haplochilic type of stomata, and they are present on the epidermis. So we can see in this diagram, so these are the haplochilic or sunken stomatas in case of Pinus leaf. So each stoma opens internally into a substomatal cavity and externally into the respiratory cavity or vestibule. So ये जो stomata है internally it opens into the substomatal cavity और externally जो gases exchange होता है that is through the respiratory cavity और vestibule भी कहा जाता है उसे. This epidermis is followed by the hypodermis, which is sclerenchymatous and is made up of thick walled dead cells. So hypodermis is present below the epidermis and is few layered, made up of thick walled cells. These are sclerenchymatous cells and it is well developed at the ridges. जो ridges हैं वहाँ पे ये well developed होती है. Then this hypodermis is followed by the mesophyll cell. और जो इसका mesophyll होता है Pinus के needle का, it shows some special peg-like ingrowths. So it in between the hypodermis and endodermis is present this mesophyll tissue. It performs the function of photosynthesis, and this cell and the cells of mesophyll are polygonal in shape and are filled with chloroplast, and there are many peg-like infoldings. So we can see here, so these infoldings are present in the mesophyll cells of Pinus needle towards inner side of the wall of mesophyll cell. There are few resin canals which are present in the mesophyll adjoining the hypodermis. So यहाँ पे resin canals भी present हैं and their number is variable, but generally they are 2 in number. वैसे इसका number vary करता है, but generally they are 2 in number. Then the cortex is followed by the endodermis, which is a single layer of parenchymatous cells and contains prominent Casparian strips. So endodermis is single layer with barrel-shaped cells and a clear-cut and bears clear-cut Casparian strips. Inner to this endodermis is presence of multi-layered pericycle that may be parenchymatous or and contains a T-shaped band of sclerenchymatous cells जो separate करता है दो vascular bundles को. So pericycle is multi-layered and consists of mainly parenchymatous cells and some sclerenchymatous cells which forms T-shaped girdle, and that girdle separates the two vascular bundles. दो vascular bundles को ये sclerenchymatous patch separate करता है. On the either side of vascular bundle there is a presence of transfusion tissue, and this transfusion tissue is made up of 3 types of cell, which are parenchymatous cells. As we know, this most of the pericycle is made up of parenchymatous cells, और इसका जो function है, it mainly helps in storage of starch. The next are albuminous cells. ये जो albuminous cells है, these are rich in cytoplasmic cell phloem in the parenchymatous pericycle and are called albuminous cells. They also helps in storage of starch. The next are tracheidal cells, and these tracheidal cells are elongated tracheates-like cells which are present adjacent to the xylem and are called tracheidal cells. And these cells carry water and minerals salts from xylem to the mesophyll tissue. जो xylem है वहाँ से ये mesophyll tissue को conduct करने में help करता है. So this transfusion tissue helps in lateral flow of nutrients in the absence of lateral venation. As we know, ये जो needle है, ये foliage leaf है, इसमें lateral veins absent होती है. So this function is carried out by these this transfusion tissue. So it helps in lateral conduction of water and nutrients in the leaf of Pinus. Next are vascular bundles. So vascular bundles in case of Pinus needle are conjoint, collateral and closed type. और ये दो vascular bundles center में present होते हैं which are separated by that T-shaped sclerenchymatous tissue. So two conjoint, collateral and closed vascular bundles are present in the center of needle which are separated by sclerenchymatous tissue which is present in the center. So in this diagram we can see here, so this one is this sclerenchymatous tissue which separates these two vascular bundles. So in these vascular bundles, xylem lies towards the angular side which is inner side. So xylem अंदर की तरफ present होता है, xylem tracheids, and phloem lies towards the upper or convex or outer side of the needle. So this upper side, there is presence of phloem. Sometimes cambium may be present in the needle, but mostly the vesicular bundles are closed in case of pinus needle. So this is all about the internal structures of pinus. So this was all about today's discussion about pinus. We have discussed its external morphology and internal structures. So hope you will get some idea from this presentation. If you have any questions, queries and any suggestions, you can give it in comment section. And thank you. Have a great day.